All right, welcome. We're about to dive into this incredible world that exists inside every single one of us. A world that's run by these tiny microscopic machines. Now, these aren't made of metal or silicon. Nope. They're made of protein, and they are literally the engines that drive life itself. So let's get right into it.
You know, this isn't science fiction. Every second, trillions of these little machines are building, powering, and defending your body. We call them proteins, and their story, well, it's one of the most elegant and, honestly, high-stakes dramas in all of biology. Just take a look at this thing. It's called ATP synthase. This is happening inside your cells right now, spinning just like a microscopic water wheel. It is a precision-engineered motor that is generating the energy you're using just to watch this. I mean, this is the level of complexity we're talking about, and that's just one of thousands of these things. So if our bodies are just packed with these incredible little motors, how are they built? Well, that's what we're going to get into in our first section, Life's Tiny Machines. You see, proteins are the absolute workhorses of the cell. They do everything. They give our hair its structure. They carry oxygen through our blood. They speed up all the essential chemical reactions. But here's the thing. As powerful as they are on their own, a lot of the most important proteins, they don't work alone. They team up. And that brings us right to section 2, assembling the protein team. Because in the world of proteins, teamwork can unlock abilities that a single protein could never ever dream of having on its own. And this slide, this just perfectly shows you the difference between working solo and working as a team. So on the left, you've got myoglobin. It's a lone worker storing oxygen in our muscles. Simple enough. But on the right, that's our star for today, hemoglobin. You can see it's a 4-part team, and this team structure is exactly what makes it a master of hauling oxygen through our entire body. Now, this whole team-based assembly thing, it has a technical name, quaternary structure. But seriously, don't let the jargon scare you. All it means is you're arranging individual protein parts, we call them subunits, to form one big functional machine. It's just like putting all the different parts of an engine together to make it run. So why bother teaming up? Well, the advantages are huge. For one, it's genetically efficient, kind of like using the same type of brick to build a giant wall. But it also allows for this truly amazing thing called cooperativity, and this is the secret sauce. For hemoglobin, it means that when one member of the team grabs an oxygen molecule, it sends a signal to the other parts, basically telling them to get ready. It makes them even better at grabbing oxygen too. All this teamwork basically turns hemoglobin into a super smart delivery system. Think of it like a molecular switch. So in the lungs, right, where there's oxygen everywhere, it flips into this relaxed state to eagerly grab it. Then, when it gets to your tissues that are starved for that oxygen, it switches its shape again to a tense state, letting the oxygen go exactly where it's needed. This perfect delivery system, it's only possible because of that 4-part team. Okay, but wait a minute. This all leads to a massive question. How do these parts, these long, stringy chains of chemicals, how do they know how to fold themselves into these perfect, complex shapes in the first place? This, my friends, is the miracle of the fold. And this brings us to maybe the single most important rule in the entire world of proteins. Shape is everything. It's like a key. If a key has the wrong shape, it's just a useless piece of metal, right? Well, for a protein, the wrong shape means the machine is broken. Its form dictates its function, period. Think about this for a second. A protein starts out as just a long, floppy chain. The number of ways it could possibly fold it's astronomical. I mean, it's so vast that if it tried to test out every single possibility, it would take longer than the age of the universe. Seriously. We call this Leventhal's paradox. And yet, inside our cells, this whole process happens in milliseconds. How is that possible? Well, scientists like to picture this process as a folding funnel. Up at the wide top, you've got this unfolded, high-energy, totally chaotic protein chain with millions of possible shapes. As it starts to fold, it rapidly rolls down the sides of this funnel, always looking for that one single, most stable, low-energy shape at the very bottom. That one perfect structure, that's its final destination. Now, the cell is a super crowded, messy place, right? So sometimes the folding process can go a little sideways. A protein might get stuck or fold the wrong way. And when that happens, the cell calls in the quality control specialists. These helper proteins are called molecular chaperones, and you can think of them as a little isolation chamber. They grab a misfolded protein, pop it inside, giving it a protected, private space to try folding again, And then once it gets the shape right, they release it back out. It's like the cell's own built-in repair shop. Pretty cool, right? But what happens when this beautiful, elegant process, this precise molecular origami, fails? Well, that takes us to our final section, the absolutely catastrophic consequences of when good proteins go bad. So let's go back to our friend hemoglobin. It's built from 574 amino acids. What do you think happens if you just change one of them? Just one tiny little alteration in the entire blueprint. Well, the consequences are devastating. And this is what happens. On the left, you see healthy, round, flexible red blood cells that can flow easily through your veins. But on the right, those are the cells of someone with sickle cell anemia. That one little change makes them rigid, and they get distorted into that crescent shape. Instead of flowing, they get stuck, clogging up blood vessels and causing terrible pain and damage. This slide shows you exactly why the cells change shape. That single wrong amino acid acts like a tiny piece of molecular Velcro, creating this sticky patch on the hemoglobin molecule. This makes all the proteins start clumping together into these long, rigid fibers that literally stretch and work the entire cell from the inside out. So you see, sickle cell anemia isn't a disease of not having hemoglobin. It's a disease where the hemoglobin folds and assembles incorrectly. It is a textbook story of protein misfolding. And believe me, it is not alone. Just look at this list. Alzheimer's, Parkinson's, Huntington's, ALS, even mad cow disease. All of these devastating conditions are, at their very core, protein misfolding diseases. Yeah, they're different proteins, they affect different parts of the body, but they all share the same fundamental error, a mistake in the fold. In a lot of these diseases, the misfolded proteins don't get fixed or cleared away. Instead, they just start clumping together into these sticky, tangled messes called amyloid fibrils. These are the infamous plaques that are found in the brains of Alzheimer's patients, a complete and catastrophic failure of that molecular origami. You know, when you really step back and look at the big picture, the takeaway is just breathtaking. Our health, our very lives, depend on trillions of proteins folding perfectly, every single second of every single day. From the oxygen in our blood to the thoughts in our head, it all comes down to the fold. And that leaves us with one last profound question. We're finally starting to understand this incredible process in breathtaking detail. So as we continue to unravel the secrets of the fold, the ultimate challenge is this. Can we actually learn to intervene? Can we learn how to correct the mistakes? And in doing that, rewrite the story for millions of people affected by these diseases. That right there, that is the hope that drives science forward.